Hi everybody. For this video we're going to go through different types of receptors, sensory receptors, and then we'll look at some special sense pathways. So this is found in chapter 6 of the Sherwood textbook. Get it to advance. There we go. All right. So this is part of the peripheral nervous system, right? So again, the peripheral nervous system has nerve fibers that are going to carry information between the CNS, right, and other parts of the body. So basically, other parts of the body can sense the internal or the external environment, and then through these peripheral nerves, bring that information to the spinal cord and or the brain, right, in the CNS. This is when it this is very important for maintaining homeostasis as well. So we're really at this point, we're looking at the afferent division. Afferent meaning bringing information to the central nervous system. Okay. And then, of course, once it gets processed there can be an efferent output. But we're not going to go through that in this video. Okay. Remember in your lessons of the brain, processing of sensory input happens by the reticular activating system in the brainstem, and that's really critical for the cortical arousal and then the consciousness. So just kind of keep that in mind as we go through this. And basically what we're doing here is we're using this processing, this central processing of sensory information that helps us to perceive the world around us, right? And that all starts with, at the beginning of those afferent nerves, receptors. So we'll look at the different types of receptors that we have in order to be able to process the world around us. And one thing just to note that sensory stimuli can also have a very big impact on our emotions, and many of our stimuli go that neural processing goes through the limbic system, which is what controls our emotions too. So it's just kind of interesting. We're going to keep it really simple. This can be very, very complex, but again, for our class, we'll keep it very simple. Okay, so we're going to start with the receptors. So just remember that we're using receptors in a different way than we did when we were looking at the synapses between neurons, right? So receptors that are on the cell membranes of our cells, like they are at our synapses. Also, if a hormone binds to an effector cell, a target cell, those are receptors that are embedded within the cell membrane. These, although they have the same word, they're a little different. These are receptors. They are located at the peripheral endings of afferent neurons. So they're larger. They're not just in the cell membrane. They're actually part of the end of the afferent neuron. And if you took anatomy, then you looked more at the structure of different types of receptors. Okay. For us, these are the structures that detect the stimulus, whatever it is that our body is perceiving. Okay. And we'll see there's a specific type of receptor that can be stimulated by specific types of stimuli. We're going to look at that in a minute. And basically, the job of the receptor, this is very important. It converts these different forms of energy into the electric signals or the action potentials, because that's the communication through the nerve that we learned about. So if somebody touches you, that's a pressure, physical pressure they're pushing on you. The receptor then, that specific type of receptor, can turn that pressure into an action potential. Okay, and you learned already how that happens. Okay. So, very important to know these different types of receptors. So again, receptors are specific. They have a specific modality or a specific type of stimulus that they respond to. They don't respond to other stimuli. So for example, we've got photoreceptors. These are stimulated by visible wavelengths of light, at least in our eyes. They do not respond to physical pressure being pushed against them, okay, to a point. So let's just go through these real quickly. Mechanoreceptors are sensitive to mechanical energy, meaning movement. It can be that, again, pressing against something. It could be fluid pressing against the receptor. So, one type of mechanoreceptor is the baroreceptor that we find in our blood vessels. Thermoreceptors respond to heat or to cold. Osmoreceptors respond to changes in the concentration of fluids in our body fluids, right? Or changes in osmotic activity. So we will have osmoreceptors in our blood vessels as well to detect changes in the concentration of substances in our blood, in our plasma. Okay, so these we'll look at these later in the semester. Chemoreceptors are sensitive to specific um, chemicals. So they include receptors for smell and taste. But even within our body, internally, we have chemoreceptors that can detect oxygen and carbon dioxide concentrations. So we'll see that when we do the respiratory system. And then this is an interesting one here, nociceptors. These are specific to pain. So they get sensitive to when there's going to be tissue damage or distortion of the tissue, right? So they might work in concert with a thermoreceptor. You might detect the thermoreceptor will detect things are getting warm, but if it starts to get too warm and it's damaging the tissue, that means it's too hot, then the nociceptor kicks in. So it's primarily a protective mechanism that's meant to bring that conscious awareness that a tissue is being damaged or that tissue damage is about to occur. We have the ability to store painful experiences of memory. So you might remember yourself or your children, you tell them not to touch the hot stove, but they do anyway. And after that, they never touch a hot stove again, right? So they have the ability to store and learn from that. Okay. So what are the receptors doing? Again, they're able to detect that stimulus and then they're changing it into an electrical potential. And they do that by that change in the membrane permeability. Okay. And that change in membrane permeability could be the influx of sodium ions. It could be stimulating a G protein cascade, something like that. So they can work in different ways. It just depends on the specific type of receptor. Okay. And again, there's going to be the magnitude of the receptor potential. How strong is it? Okay. And so that's also a type of graded potential. That depends on the intensity of the stimulus. Okay. And so if it is sufficient magnitude, like a graded potential, then it will stimulate the action potential. There has to be a threshold. And so you might be thinking now, it might sound obvious that there's going to be a different threshold associated with different receptors, different sensory receptors, and their neurons. Okay. So this is just what that sequence might look like. So here we have an afferent neuron. Okay, at the end of the afferent neuron would be the sensory receptor. So this is where the stimulus would start. Okay, again, graded potential, if it's strong enough, here's the site of initiation of the action potential. The action potential would run down the cell body and it's going to synapse with an interneuron. And depending on, this might have a different threshold, this interneuron. So you might need a lot more input than just one cell, one receptor, right? It just depends on what it is, what our bodies, and it could be variable individually as well. Okay, so again, producing another action potential, which would then produce an action potential on an efferent nerve to get us to respond to. So again, if this was heat, okay, and maybe it's something that feels warm and our brain determines, oh, we like that warmth because we were cold, it might get you to actually try to hold and get closer to that heat source. Okay. So again, receptors, they can be specialized ending of the afferent neuron. it might be a different cell, actually a separate cell, like in the case of the photoreceptors. We'll look at the eye and we'll see that in the photoreceptors, those are actually separate cells. Okay. All right. Let's go to the next slide here. So this one is just showing you how the magnitude of the receptor potential can change the frequency of the action potential. So here it's showing you that the rate of the neurotransmitter release at the afferent terminals, that can affect the frequency of action potentials at the afferent fiber. The afferent fiber is the same thing as the axon of the afferent neuron. Okay. Remember, sometimes we call things fibers because they're long, but they're referring to the axon. Okay. Um, there could be the receptor potential. How strong is the receptor potential, right? And that can affect the action potential. Okay, so a stronger receptor potential or a graded potential can cause the action potential firing faster. Okay. And then the magnitude of the stimulus can affect all of these, right? So if you're poking someone kind of lightly, you'll get this type of graded potential, which will stimulate just a few action potentials. There's a little time in between them. Your brain then will interpret that as a light touch. But if you press harder, right? You get a stronger stimulus, which you get a stronger potential at the receptor, which is going to cause the action potentials to fire more frequently. Okay. Okay. This you'll learn more about in the lab if you're taking the lab, but I just want you to realize that receptors may adapt either slowly or rapidly to any type of sustained stimulation. And so because of that, they're classified as either tonic or phasic. So it's either a rapidly adapting receptor or a slowly adapting receptor. So tonic receptors adapt slowly or they don't adapt at all. And these are things where you want constant input. So like our muscle stretch receptors, joint proprioceptors, you want to know what position the body is in. Okay. You don't want to lose that input. So, but you must have continuous input about the degree of your joint position and things like that. A phasic receptor is a rapidly adapting receptor. So it doesn't respond to a maintained stimulus. So that's like you'll do an experiment. I believe you'll still do it in the lab. And you can try this at home. Put a penny or a small coin on your hand and you'll feel it. But look away, think of other things. You'll stop feeling it after a while. Those are phasic receptors. Okay, and so a good example there is the tactile or the touch receptors of the skin. Okay. Tonic and phasic. Okay, so somatosensory pathways. This is what we're going to get into. Somatosensory is about, remember, that is what goes to the parietal lobe, what we're feeling usually on the skin surface. Right? And so I like this. If you follow the blue neurons, right, that's the sensory pathway. So you're going to have some type of sensory receptor on the skin. This could be touch, this could be temperature, right? Anything like that. It's going to stimulate then the afferent neuron, right, which is going to continue up, pass through the spinal cord, through the dorsal side, dorsal root ganglion, into the dorsal part. Then it's going to go up the spinal cord through the ascending tracts. You'll have this sensory axon will synapse with another neuron, which then passes through the thalamus and then on its way to the somatosensory cortex in the parietal lobe. Okay, I want to make sure when you tell me what part of the brain a stimulation occurs or the processing occurs, you need to tell me the specific part of the cortex and what lobe. So this is the somatosensory cortex of the parietal lobe. That's where we determine touch and heat and things like that, the somatosensory pathway. Okay. So I also wanted to show you that in all these pathways, you're going to just see a chains of neurons that are synaptically interconnected with one another, and they produce a particular sequence to achieve the processing of the sensory information in the particular part of the cortex of the brain. Okay. All right. And a note on perception. So, perception is what we mean by conscious interpretation of our external world. And that's all derived from the sensory input. Okay. This is the pattern of nerve impulses that are delivered to the brain. But what's really important to realize is that this sensory input does not really give us a true reality of what is out in our environment, because we can only perceive something if we have the ability to perceive it through the receptors that we have, right? So we only have a limited supply and type of receptors. And I put this picture of the flower here to give you an example. So we have photoreceptors that are able to see the visible light spectrum, right? We cannot perceive UV light. Other organisms can, like butterflies. So we see the yellow flower. A butterfly sees this pattern, this white and pink pattern, because that's the UV light reflecting off of it. It has receptors for UV determination. So its world looks very different than our world. Okay. And then also realize that our cerebral cortex manipulates the data as well. So sometimes what it's doing is it's going through our memory stores, and when you see something, do I know what this is? And it will choose something from our memory, our experiences, and interpret it in that way. And sometimes it can be quite false. I have an experience. I was mountain biking with my husband, and from a distance, I saw an owl on a log. And I'm like, look at that owl on the log. That's so cool. It's just sitting there. And he's going, where? Where? Well, it turned out it was just a part of a broken limb sticking up. But the pattern, my brain first interpreted as an owl. It wasn't an owl, it was a tree limb. So that's perception. And another note on nociceptors. You know what? I think maybe I did say this already. Nociceptors, so I did say they're primarily a protective mechanism. I think I meant to erase this slide because I said this already. But we can store painful memories. And again, when the nociceptors actually get stimulated, it elicits perception of pain. Okay, I got stuck in the middle there. Special senses. So what we're going to do now is we're going to look at, we saw the somatosensory perception, but now we'll look at some of our special senses. I'm not going to go through much of the anatomy because you'll do that in anatomy class. This is more about the sequence of neurons, starting with the receptor and the sequence of neurons and where they get processed in the brain. Okay. So you're going to know the receptor and the pathway to the appropriate area of the cerebral cortex. We'll start with the eye. So your eyes are the special sense organ to be able to detect light, to be able to detect the visible light spectrum. Okay, and it does that through the layer of tissue on the inside of our eye called the retina. That's where the photoreceptors are located. And those specific photoreceptors are called rods and cones. So those photoreceptors, the rods and cones, they get stimulated by the light, and they convert that light energy into nerve impulses or action potentials. Okay, so again, I'm not going through all of the anatomy of the eye. We'll do this in anatomy. I'm talking about, so the eye on the inside is fluid filled. If we take out the fluid, the tissue that's up against that fluid is called the retina. That's all of this. Again, that contains the rods and cones. There's an area where we have a concentration of cones. That's called the fovea. So this is where we try to direct our light onto this area where we get the highest vision acuity. Okay, those photoreceptor cells will synapse with other cells, I'll show you in a series, which then form the axons of those cells form the optic nerve. So they all kind of line the inside of the eye here and they come together forming the optic nerves. These are bundles of axons. Of course, there's blood vessels in there as well. But this spot here where all those axons come together to form the nerve, that's called the optic disc. So any light that bounces on the optic disc, there are no rods or cones there, so it's what we call our blind spot. You'll see that in just a minute as well. Okay. So here, let me show you. This shows you, here's a source of light. Light just kind of radiates out in all directions. It comes to our eye. And so we have the cornea and the lens, which helps to bend the light. And a healthy eye will bend the light onto that fovea. Again, the point where we have a high concentration of cones. So that's where we get the highest visual acuity. If we're not able to do that, the light will scatter around our retina and things will look blurry to us. Again, I'll let you learn about that in anatomy because you go through these different layers of the eye and why that happens. Okay. So lights get focused on the retina. Again, we're going to see the stimulation that stimulates eventually our optic nerve, which then goes to our occipital lobe. So if we just look at the retina itself, take a little cross section there, this is what we would see. So over here, this is the front of the retina, which means also the front of the eye. So light is coming in this direction. Okay. Then the layers of the retina, we have several cell layers, and we're going to be concerned of these three layers. Here are the photoreceptors. So that's at the back of the retina. So light stimulates, goes through, just kind of passes through, bypasses, I should say, these cells. So when we talk about the stimulation, we're going to start with the photoreceptors, which are the rods and cones. Okay. These then are going to stimulate an action potential on the bipolar cells, which will then stimulate the ganglion cells, which then form the axons or the fibers that eventually become the optic nerve. So all of these, all along the lining of the retina, these form these cords, right? These bundles of axons, which come together and form the optic nerve. Okay. If you look into an eye, here's what I mentioned, the blind spot. So that's where the optic nerve is actually forming. So again, no rods and cones there. So anything that hits there, you won't actually see it. The macula lutea, that is a structure that is actually the area that is immediately surrounding the fovea. So when an eye doctor uses a scope and looks in there, she can see where the fovea is based on where the macula lutea is. All right. So, one thing just to add, macular degeneration, this is when the tissue in this area starts to degenerate and it affects your vision, and it's one of the leading causes of blindness. I'm just going to leave that at that. All right. The next, so what we want to do now is just kind of go through that process. So again, light is going to come through here. It's entering the retina. And the retina consists of all these cells, but it's going to bypass all of these cells, okay, until it hits the rods or the cones. Cones give us color, visual acuity. Rods give us black and white. So our cones don't really function well in nighttime, okay? Our rods do. That's why at night we don't really see a lot of color. Everything kind of looks black and white. Okay, and that has to do with different types of pigment that's associated with these. Again, I'm not going to go through that. But the light stimulation is going to stimulate the ends of the rod or the cone. Okay, the rest of the cell contains this organelles, right? And then at the ends, toward the front of the eye, we get the synaptic terminal. And that then is going to stimulate the next cell in the series called the bipolar cells. Okay, so again, it's here, the end of the rods and cones, that actually detects the stimulus. All right, let's go to this one. So here's that sequence of events. So light comes in, there's your light. It activates a photopigment, which again, it can either activate the rod or the cone, depending on which photopigment. Don't worry about how we distinguish that. But that's actually going to open its sodium channels. Okay, so there's your polarization. And if you read details in your textbook, it will talk about sometimes it gets hyperpolarized, sometimes it gets depolarized. Again, that has to do the difference of the stimulus, whether we're in bright light conditions or dark. You don't have to know that. So that's why I just wrote polarization of the photoreceptor cell. Okay, and that starts your receptor potential, which is like a graded potential. We get a neurotransmitter release, right, which stimulates the bipolar cell. The bipolar cell, if it's stimulated adequately, it will release a neurotransmitter which stimulates the ganglion cell. Okay? Then the ganglion cell will form that optic nerve. Now we'll see where that goes. Here we are. We just kind of slice through the brain. You can see the eye. Here are the optic nerves. Now, your optic nerves will cross. Any information coming from the medial portion of the eyes, those nerves cross to the other side of the brain. On the lateral portion of the eye, those axons forming the optic nerve stay on that same side of the eye. Okay, so we get a little cross here. So what I want you to know is after the ganglion cells, which form the optic nerve, right, then that comes into the optic chiasm, okay, which then forms the optic tract, which then synapses in the thalamus. I circled thalamus here because that's all you need to know is the word thalamus. Okay, we synapse in the thalamus, go back and review the brain. That's the relay center for a lot of sensory input. But now those neurons will travel back to the occipital lobe of the brain, where it stimulates the visual cortex in the occipital lobe. Okay, that's where we get conscious perception of what we're seeing. That's the eye. Okay. So here I just put the visual pathway for you so you guys can know exactly what I want you to know. So I'm not going to read through this. I think you can read through it. Go ahead and pause if you need to do that and write that down. The next one is the ear. So the ear really has three parts. The external ear, these are your pinnae. What that does is that grabs sound waves or collects the sound waves. It's going to send it to the middle ear where we have bones, which are going to enhance those sound waves. And then that's going to send it to the inner ear, which is going to move fluid inside of the ear. And that fluid is going to move a tissue, which will then rub up against the receptor cells. So these are going to be mechanoreceptors. Okay. So the inner ear is where the sensory systems are located. The cochlea, again, contains these mechanoreceptors for that conversion of sound waves into nerve impulses, which then make the hearing possible. We also have something called the vestibular apparatus. This is for equilibrium. I'm not going to cover equilibrium in this unit. Okay, we'll stick with hearing and the cochlea. Again, you learn about the anatomy in anatomy class. I'm not going to go through too much of that. But one thing I want to mention, so sound waves are actually traveling vibrations of the air. So if all these little dots, these are like air molecules, okay, and they're uniform when there's no sound traveling. If we use this tuning fork and bam, you hit it, what that does is it compresses those sound waves. It causes those sound waves the air molecules to move around, and it actually then travels in a wave. That's what our ear pinnae are collecting and funneling into toward our inner ear. Okay. Um, so again, so here's the ear pinnae of the external ear. Sound waves are going to get because sound waves, just like light, just kind of travel, they scatter. So we have a funnel to collect as much as we can, and it funnels it into the external ear, and it starts to vibrate this tympanic membrane, which then vibrates these bones, the ossicles. Okay? And what it's doing is it's enhancing that mechanical movement of the sound waves. Okay? Then there's an oval window right in front of the cochlea, because the cochlea is fluid filled. Okay, and so when that last bone, the stapes, vibrates the oval window, it then vibrates the fluid, and now the fluid now is vibrating in there. And when the fluid is vibrating in there, that then stimulates tissues which are going to run up against those mechanoreceptors that are inside the ear. Okay, here's the cochlea, here's that oval window where the stapes is closing it off. So when that vibrates, tympanic membrane vibrates these ossicles. Here's the fluid compartments. And then inside, these are the receptor cells that also span throughout the cochlea. They're here. And there's a membrane. So as the fluid here starts to vibrate all these membranes, that rubs up against, here are the receptor cells. Okay, we call them hair cells because they have little cilia that stick up, so that's rubbing them, causing an action potential, or actually a receptor potential, graded potential, and then an action potential. Okay, so I thought this was real nice. This kind of gives you that step by step. We have sound waves, vibrates the tympanic membrane, vibrates the middle ear bones. That's all I'm requiring you to know for this unit. You should learn in anatomy the names of those bones. Vibrates the oval window, vibrates the fluid inside of the cochlea, right? Vibrates the basilar membrane, which is the membrane that's right up against the receptor cells. You know that those are mechanoreceptors. Okay? Causes the graded potential or the receptor potential, which changes the rate of the action potential of the auditory nerve. And the auditory nerve goes to the auditory cortex of the temporal lobe. Okay? And that's hearing. Let's go into chemoreceptors. Chemical senses come in the form of taste and smell. So we stimulate receptors that induces these chemoreceptors, and they make you either feel pleasurable or objectionable sensations, right? So it helps us to either seek that out or avoid it. Okay. Usually things that are bad for you smell bad. Things that are bad for you taste bad, usually. And so we have an innate response to want to get away from things that taste or smell bad. Okay. Let's do taste first. So for taste, again, we have chemoreceptors, and they're housed in the taste buds that are within the papillae on our tongue. So all these little bumps are called the papillae, and the taste buds are embedded in there. I'll show you in another picture. We've got some in the oral cavity in the throat. They're probably more active as infants, and then they are not as active as an adult. But the ones on the tongue are. And what we'll see is that in order for a taste molecule, a chemical, it has to dissolve in fluid, which our mouth is wet, and it will go down into a pore where it finds the taste receptor. So that's what this is. This whole thing is the taste bud. Here's the pore, right? These are the taste receptor cells. Okay, and here's where those chemicals combine. So anything on the surface of the tongue, that chemical will dissolve in the fluid and go down into the pore and stimulate the taste receptor cells. And there's the sensory nerve fibers. Okay, so from there, our sensory nerve fibers, I erased some of this stuff here. I don't care that you know what these are labeled. Okay, these are just the cell bodies of these neurons. Okay, And they come up through the brain stem, um, all the way up to the thalamus, where then it goes to the primary gustatory cortex, which is actually in the insula. So go back to the brain unit and see that's kind of that folding of where the parietal lobe and the temporal lobe kind of fold inward. That's an area called the insular cortex. So here's the list. Chemoreceptors in the taste pore and on the taste receptor cell get stimulated. Um, it sends that sensory information to the nerve fibers. Looks like I dropped a Y there. I'll change that. It's then those nerve fibers are sensory nerve fibers, afferent nerve fibers, right? Synapse in the thalamus to the primary gustatory cortex of the insula. I wanted to put this in here just so you know that our receptors, we have different receptors that can be stimulated by different types of chemicals. So these are the different tastes that we have. Okay. And one of the things I did want to mention here too is that our taste can also be affected by other senses in our near our mouth. So olfaction is one that can affect taste, and then also the pressure in our mouth. So the consistency of food can also give us um, a different um, quality of taste, right? I always thought I hated oatmeal until one day I put too much milk in it or water, whatever it was, and it was real thin and I liked it. So I didn't like lumpy oatmeal. I like real thin oatmeal. So I thought it was a taste thing, but it's not. Okay, last one is smell, or that's the process of olfaction. So we have olfactory receptors, which are chemoreceptors that are in our nose, and they're basically specialized endings of these neurons. And since we always have, we blow our nose, we have things going on in there, those can get worn away, but we have the ability to regenerate them, okay, with these cells called basal cells that you'll see in a minute. Okay, so the olfactory mucosa is in the lining of the nasal cavity, and we've got these three cell types in here. The basal cells are the ones, again, that are stem cells that help us regenerate. Okay. And that here, as you can see, inside of the nasal cavity, again, just a 3 centimeter square space is where all of our chemoreceptor cells are, or olfactory cells. So remember, they're olfactory receptor cells. Remember, they're chemoreceptors. Okay, it projects through the bone. This is the ethmoid bone. So here's the receptor, like I said, the receptor cell, and the end of it projecting through the mucous membrane are the cilia, and this is where the chemicals can bind to it. And again, the chemical has to be dissolved in fluids, so that's why we have mucus in our nose. And that one of the reasons, and that helps that to bind to the receptor cells. That will send a graded potential through the receptor cell, which then enters up through an afferent nerve fiber into which is the olfactory nerve, and into the olfactory bulbs. And you saw those in anatomy as well, part of the brain. Okay, and let's go in there. So here I have it listed for you. We got through here's the olfactory bulb, one of them. We've got two, but you can see all of these nerve endings from these olfactory receptors. Synapse with the glomeruli and the olfactory bulbs, and this is what sorts the odor molecules. So again, we have different molecules based on experience of different odors that we smell, and so that'll get sorted out. So certain smells go to a certain glomerulus, which will then stimulate one of these mitral cells, which then helps to form the olfactory nerve. Okay, so if the action potential goes on here, then after the mitral cells, we can have two routes. One route goes through the limbic system and then to the primary olfactory cortex of the temporal lobe. And that's where the temporal lobe can coordinate that smell with behavioral reactions. I'll leave it up to your imagination what a behavioral reaction might be. The other pathway is those mitral cells can split, and those axons can go through the thalamus, which then go to the frontal lobe, to an area called the orbital frontal cortex, which is actually just above our nose, just above that ethmoid bone, as your frontal lobe comes down. And that's where we get the conscious perception of smell. All right, that's it. Thank you, everybody. Hopefully that helps you understand receptors better.